Hello, everyone. We are pleased to have you join us for Cardinal Health Lab Exchange. Welcome to today's live broadcast. Carbapenemase-producing GNB, the nightmare bacteria, are here to stay. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's webinar. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by the American Society for Clinical Laboratory Science, ASCLS. ASCLS makes a positive impact in healthcare through leadership that assures excellence in the practice of laboratory medicine. For more information, please visit www.ascls.org. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. Following this presentation, head over to the Community of Learning in the Lab Exchange lobby to have the opportunity to engage in live chat and have more of your questions answered on the spot about Carbapenemase-producing GNB, nightmare bacteria that are here to stay. I would now like to introduce today's speaker, Nicholas Moore, MS, MLS, ASCP, CM. Nicholas Moore is an instructor in the Department of Medical Laboratory Science at Rush University Medical Center. Nicholas earned his Master of Science degree in Clinical Laboratory Science at Rush University in Chicago, and upon graduation, he worked for 2 years in the microbiology and core laboratories at Rush University Medical Center. In 2011, he accepted a position as a laboratory director in a long-term acute care hospital, LTACH, in Chicago. While in the LTACH, he was involved with investigators in a CDC-funded study that involved the implementation of an infection control bundle to reduce Klebsiella pneumoniae Carbapenemase KPC transmission and infection in the LTACH setting. He is currently an instructor in clinical microbiology in the Department of Medical Laboratory Science at Rush University Medical Center and is a doctoral candidate. His research efforts focus on KPC and other antimicrobial resistance mechanisms among gram-negative bacilli. I will now turn it over to Nicholas Moore for his presentation.
Thank you, Judy, and thank you everyone for attending today. I was very honored to accept the invitation to come and give this presentation. So we'll go ahead and get started. I do have just a few disclosures that are there. I'm not going to be talking about anything commercial today related to any of these disclosures, but you have them there for housekeeping purposes. Here's today's objective. So by the end, I hope everyone is going to be able to discuss laboratory methods that can be used to detect clinically important carbapenemases, to identify patient-level risk factors for KPC colonization in long-term care settings, and then we'll end by discussing recommendations and strategies for controlling CRE in healthcare facilities. So I'd like to start with just a quick poll to each of you in the audience about what method your clinical laboratory might use to detect CRE. So if you want to take about the next 15 to 20 seconds to answer this question, that would be great. So a couple of different options. You could do, your lab might do a broth enrichment or some culture-based mechanism. They could use a chromoger to screen for CRE carriage. They might just use a phenotypic method like the modified Hodge, a PCR test, or if you don't know, or if your lab doesn't routinely detect CRE in clinical or surveillance cultures, that's fine too. So a box should have popped up and you can select which option best fits what your lab is doing and then click the submit button. Otherwise, if you don't wish to answer, I think there's an X in the upper right corner of that box that appears. All right, so let's see what everyone's responses were. So it looks like about, for most people who responded, if they do a test, it looks like you're performing a phenotypic-based method, like the modified Hodge test, which is one of the methods that's been well published and publicized and discussed about. And then about an even number of you don't know if your lab is currently doing something to screen. And then very few people use PCR, and that's not unexpected. And then even about the same amount may use some type of chromogenic agar. So we're going to discuss each of these today in a little more detail as we go on. So let's go ahead and just get started. I like to give kind of a brief overview just to kind of set the stage. So here in the US, there are approximately 2 million healthcare-associated infections, or HAIs, that occur annually, and that costs the United States an extra $45 billion in healthcare spending. And as we have seen increasing rates of antibiotic resistance, especially among the gram-negative bacilli, and coupled with a lack of development of new antibiotics from pharmaceutical companies, this has really created a very dangerous and important public health crisis. And this is a nice paper that was published about 5 or 6 years ago in the Journal of Clinical Infectious Diseases, looking at actual, hard cost, both hospital costs and then societal costs in patients who have infections caused by antimicrobial resistant organisms. And the kind of 3 takeaways from this paper are that patients with these antimicrobial resistant infections have increased rates of mortality compared to patients with non-antimicrobial resistant infections. And these patients also tend to stay in the hospital for about an extra week to 2 weeks, so anywhere from 6 to almost 13 days of additional time spent in the hospital. And all of that correlates to about anywhere from $18,000 to a little over $27,000 additional dollars spent in the hospital care on a per patient basis. So antimicrobial resistance has a huge impact on not only the health and well-being of our patients, but on our ability to provide care for them. So in 2013, the Centers for Disease Control and Prevention declared that Carbapenem-Resistant Enterobacteriaceae, or CRE, are an urgent public health threat. CRE is a very broad family, or a broad description. So these are the Enterobacteriaceae that are carbapenem resistant. And specifically, those CRE that are KPC and that they produce the Klebsiella pneumoniae Carbapenemase, that is the most common mechanism that is encountered worldwide. I'd like to just go briefly over the beta-lactam antibiotics. These beta-lactams can be divided into 5 kind of subgroups or families. You have your penicillin derivatives, the cephalosporins, the cefamycins, the monobactam aztreonam, and then your 4 carbapenems on the right. All of these antibiotics are beta-lactams or beta-lactam derivatives. They are designed around the similar beta-lactam structure, which you can see kind of in the green shaded ring in that bottom figure on the slide. So that is the active portion of the antibiotic. The beta-lactam ring is what recognizes and binds to those penicillin binding proteins in the bacterial cell wall. And when it binds to those penicillin binding proteins, it prohibits and prevents some key enzymes from forming those cross-links and those bridges that give the bacterial cell its strength and rigidity. And by preventing that cell wall from forming, these beta-lactams exert their bactericidal or killing effect on the bacteria. Now, the bacteria are very smart and have evolved over time to come up with ways to avoid the activity of the beta-lactam ring. And one way they do that is by the production of special enzymes that hydrolyze these rings. And that you can kind of see happening on the right-hand side of the figure. where this beta-lactam ring has been hydrolyzed and opened up. If that beta-lactam ring has been hydrolyzed, then the beta-lactam can no longer recognize and bind to those penicillin binding proteins. The antibiotic doesn't prevent those transpeptidases from forming. The bacterial cell wall remains formed and strengthened, and the bacteria continue to survive. So the carbapenemases are carbapenem hydrolyzing beta-lactamases that confer carbapenem resistance among these gram-negative rods. Now, there are a lot of ways that bacteria can become resistant to carbapenems, and this is very distinct from Enterobacteriaceae and other gram-negative rods that possess efflux pumps or have alterations that lead to porin mutations, which prevent the drug from entering the bacterial cell. Now, what some people have done is taken these carbapenemase enzymes and organize them into a classification system. And this is based on the homology between the amino acid sequences, and this is the Ambler molecular classification system. So for the ones that I'm going to talk about today, your carbapenemases that fall into class A, C, or D all share a serine residue kind of at the center of this enzyme, and that's what gives that enzyme its biologic activity. In contrast, your class B enzymes, which are also known as the Metallo beta-lactamases, instead of possessing a serine molecule, they have a divalent cation like zinc in the center for their activity. This is a table just kind of going over some key points of the 3 carbapenemases that we're going to talk about today: KPC, NDM, and the newest to kind of show its face here in the US, the OXA-48-like carbapenemases. All of these are found on plasmids, which leads to their very high potential for dissemination across bacterial species and also to other patients. But they have varying levels of activity to some of these beta-lactams, and this can kind of be a clue when you look at a susceptibility report about what potential carbapenemase may be present, and we'll talk more about that towards the end. So if you look at organisms that produce the KPC carbapenemase, for example, they typically have hydrolytic activity towards all beta-lactams. So in other words, all of the beta-lactams that you might test are going to typically be resistant or have very high MICs. In contrast, organisms that produce the NDM gene are going to still remain resistant to all beta-lactams except for the monobactam estrianam. So if you're resistant to all of your beta-lactams but estrianam remains susceptible, it's a very high possibility that the organism is producing a carbapenemase that's NDM. And then the OXA-48-like carbapenemases are often the most difficult to try to identify without molecular typing or PCR methods. And that's because they have relatively weak hydrolytic activity of the carbapenems. The MICs are going to be slightly elevated or, just, but very much lower compared to KPC or NDM. And they also remain usually susceptible to your later 3rd-generation cephalosporins, so ceftazidime, ceftriaxone, and ceftriaxone, and those other later-generation cephalosporins, unless the organism is also an ESBL producer or possesses, again, alterations in the porin mutation or an efflux pump. So the OXA-48 are typically the hardest to identify just using phenotypic methods alone. This was the paper that was published back in 2001 that really described the first bacteria. It was a Klebsiella pneumoniae that produced the Klebsiella pneumoniae carbapenemase. And again, this has really become the most clinically important carbapenemase that we're seeing worldwide. Since its initial description in 2001. It has spread throughout the United States and also spread globally, and that is really due to the fact that it can be found on this very mobile transposon that can then be inserted into plasmids from a lot of other gram-negative rods. So that's really what allows for it to spread so easily. In Chicago, KPC was first identified in our region back in 2007, and it was identified in a patient who was transferred to a West Suburban medical center from a skilled nursing home in Northwest Indiana. Later in 2008, Rush, my institution, received their first patient who also came from a post-acute care facility, a long-term acute care hospital, or an LTACH in Northwest Indiana as well. So I don't know what Indiana's been doing, but they've really been introducing a lot of bad bugs to the Chicago area. And it was really with that introduction and that first patient who was recognized in Chicago that kind of really energized some infectious disease practitioners and scientists here at Rush. So we did a study that looked at how this organism was spread and introduced. So I really like this figure. It was published in this paper by Sarah Wan in 2011 in Clinical Infectious Diseases. And what you're looking at is a modified social network analysis. So when we think about social network analysis, you're typically looking at interactions between people, and in this case, instead of looking at interactions between people, we're looking at interactions between different healthcare facilities and the types of friends, if you will, that they share in common. So what we found is that in this retrospective kind of look-back study, there were 40 patients who were identified, and they are represented by the blue triangles, who were found to be KPC positive, either under surveillance or clinical cultures. A majority of these patients had a direct exposure to this red circle in the center, which is this a particular long-term acute care hospital or LTACH. And these patients spread throughout the metropolitan Chicago area to 14 different acute care hospitals, which are these kind of golden yellow circles, and also to 10 additional nursing homes, which are the kind of lightly shaded green circles. So in total, this one facility kind of played a role as an epicenter, or for this focal point of this outbreak, that gave or transmitted KPC to 40 different patients that affected 26 healthcare facilities in the metropolitan Chicago area. So I think this is a very striking figure that kind of shows just how quickly, and this was only a few months in which this occurred. And only 2 case patients in this study, patients 7 and 17, you can kind of see them in the lower right corner, were not directly linked back to this this LTACH in the epicenter of the outbreak. And again, this figure just wants to is really just here to show you again how quickly KPC, the KPC carbapenemase has spread throughout the country since its initial description in 2001 up until now. I was at an infection control conference yesterday, so my slide is out of date, of course. But as of yesterday, there have been reported cases of KPC identified in Alaska. So we should shade Alaska in green in the lower right. So only 2 states don't have any reported and confirmed cases of KPC producing CRE. That's Maine and Idaho. But again, in a relatively short time, this enzyme has spread quite rapidly throughout the country. And again, also throughout the world. I don't have any figures to show you, but it's prevalent it's very prevalent in parts of Europe and Asia and the Middle East. So why do we really need to care about KPC or other CRE? Really, this is a nice study that was published back in 2008, looking, so this was a case control study, looking at case patients who are infected with a Carbapenem-Resistant Klebsiella pneumoniae. So these are this is the bar shaded in blue, and then they were compared one-to-one to patients who were infected with a Carbapenem susceptible strain of Klebsiella pneumoniae. And the case patients were matched to controls based on their site of infection. So what we see overall is that our case patients, so those who had a Carbapenem-resistant K pneumo infection, were about 3.7 times more likely to die, 48% versus 20%, to patients who had a Carbapenem susceptible K pneumo infection. And that was just for overall mortality. And that result was statistically significant. You can see the p-value there. And then if we look at attributable mortality, so patients who died directly as a result of their infection, again, attributable mortality, case patients, 38% versus 12%. So 4.5 times more likely to die if you have a Carbapenem-resistant infection versus a Carbapenem-susceptible infection. So really, just that these bugs can spread very rapidly, and if patients develop an infection, they have very high rates of mortality associated with them. So kind of just to wrap up briefly on KPC, it's rapidly emerged as an epidemiologic important mechanism of resistance in gram-negative bacilli that are multi-drug resistant. They have very high rates of mortality. Other studies looking at the same thing show rates between 30 and 50% mortality rate if patients develop an infection. And a lot of how KPC has been able to spread so rapidly is due to this dissemination of this hyperepidemic clonal type, multi-locus sequence type ST258. So multi-locus sequence typing is just basically a way to look at the fingerprint, if you will, of the organism to give it a discernible type. Similar to PFGE, but we're actually looking at 7 different housekeeping genes that are typically readily conserved. They're amplified by PCR, and then they're looking for mutations in the amino acid sequence of these genes, and every time a new mutation is identified, it gives it a new sequence type. So my colleagues in infectious diseases like to talk about the resistance iceberg, and I was told if I was going to give this talk, I had to mention it as well. So when we think about an iceberg, there's kind of what you see on the surface of the water, which is what's floating, and this really represents those patients who are clinically infected with a Carbapenemase-producing organism. So these are patients who have some type of clinical culture. It could be blood, urine, sputum, and evidence of an infection. They could have a high white count, fever, signs of SIRS or sepsis, hypertension, rapid heart rate, but there's a documented source and clinical signs of infection. What we have to worry about, though, is everything that's happening beneath the surface that we can't see. And this really represents the patients who are asymptomatically colonized or are carriers of CRE producing bacteria in their gut. So these are patients who are rectally colonized, who don't show signs of an active infection. They don't have a high white count, they're not febrile, and really you only detect these patients if you're looking for it on a surveillance culture. And why this is so important, and why this is really the large part of this resistance iceberg, is that these are the patients who, if you don't know that they have it, they will go unrecognized, and if they're not placed on contact precautions, if they're not isolated, these bacteria can be spread to other patients in other healthcare facilities, either directly or indirectly via healthcare worker hands. So unrecognized colonization is what really, I think, led to a lot of the high rates in some of these facilities that we're going to look at. Briefly, just to go over the Metallo-beta-lactamases, or the MBLs. Again, this is your class B Carbapenemase. These, again, are dependent upon zinc in order to hydrolyze the beta-lactam. Now, interestingly, these are able to be inhibited by EDTA, as I'm sure many of you are familiar. This is an ion or cation chelator. So we can use this in a method, in a culture-based method, to presumptively identify an MBL. If we compare an EDTA inhibitor with a carbapenem, and I'll describe that here in a little bit. The first MBL was actually described in Japan in 1991. It was the BLA-IMP metallo-beta-lactamase enzyme, and since then dozens, if not hundreds, of others and variants on these other metallo-beta-lactamases have been identified. Now, some MBLs are naturally occurring and chromosomally encoded in species. We don't have to worry about them. If they're encoded in the chromosome, they're not an infection control emergency. They're not going to be spread easily between different bacterial species. We do worry about the MBLs like NDM and VIMP and IMP because they are found on plasmids and other mobile genetic elements like integrons, and those can be readily disseminated, and those are an infection control emergency. So NDM, or the New Delhi metallo-beta-lactamase, was probably the one that, if you've heard of, are most familiar with. It was first described also in an isolate of Klebsiella pneumoniae. This was found or isolated in a Swedish patient who had spent some time hospitalized in India before he returned and was repatriated to his country back in December of 2009. So again, NDM, like KPC, is found on a plasmid, but NDM tends to be a little more complex and unpredictable when you compare it directly with Klebsiella pneumoniae carbapenemase, or KPC. Like KPC, it's not restricted only to K. pneumo. We find it in other strains of Enterobacteriaceae. There have been outbreaks of NDM E. coli, as well as reports of NDM E. cloacae in the literature. But really, along with KPC, NDMs can be recovered from any Enterobacteriaceae member. This is probably something that maybe some of you might have heard about and others maybe not. But in Chicago, another claim to fame aside from the Cubs and the Blackhawks, Chicago was the site here in the U.S. of the largest NDM outbreak that's been reported so far. So this was an outbreak that was published in the Journal of the American Medical Association back in 2014 that was linked to an NDM-producing E. coli that was associated with patients who had been exposed to duodenoscopes. Those are a kind of a very fine instrument with a camera that kind of is passed through the esophagus, through the stomach, to visualize into the small intestinal tract and into the biliary tree for a procedure called ERCP, endoscopic retrograde cholangiopancreatography. Try saying that 3 times fast. But anyway, this outbreak investigation, in summary, identified 44 patients who had been exposed and had rectal carriage of NDM E. coli after undergoing this ERCP procedure in a suburban Chicago hospital. Now, what makes this outbreak a little different compared to some others that are associated with bacterial contamination of endoscopes or duodenoscopes is that public health officials, CDC experts, and the device manufacturers, when they came in and assisted this facility in their outbreak investigation, found that and were able to recover this organism after it had been, after the instruments had undergone manual cleaning as well as high-level disinfection. And the public health officials at APH and CDC were not able to identify any lapses in the cleaning procedure that the facility was using to service these scopes. So that really was another clue that maybe there was something wrong with the design of these scopes, which again came up when we started hearing in the popular press a few months back about the outbreak that affected the UCLA Medical Center. And that outbreak that affected UCLA was due to a different carbapenemase. This was the OXA-Carbapenemases family. These are your class D carbapenemases, also known as the oxacillinases, that are associated with carbapenem resistance. Now, there are several hundred types of OXA enzymes, and I've really tried to group them into 5 subfamilies. And the subfamily that's been associated with this outbreak is the OXA-48-like carbapenemase, which is found on a plasmid along with OXA-23, OXA-2440, and OXA-58. The only one that is chromosomally encoded is OXA-51, and those are intrinsic, and so far, I believe, have only been recovered and isolated in species of Acinetobacter baumannii. So the UCLA outbreak a few months back was due to an OXA-48-like carbapenemase. Back in on February 18th, the facility notified 179 patients that they may have been exposed to CRE bacteria during their ERCP procedure. Again, this was linked to these duodenoscopes. Last I checked, 7 patients had been actively infected, and 2 of whom actually did die as a result of, not necessarily attributed to their infection. These are patients who are already very chronically sick, but 2 patients did die after undergoing this ERCP procedure. So there's been, again, a lot in the popular press. You may have seen some of these are very similar headlines, and really this is kind of, again, I think highlighting why antimicrobial resistance is so important, why this is a topic that we should be discussing, and why I was glad to see last week that the federal government and the White House held a summit or a conference on this topic, and how the federal government is going to respond to not only these outbreaks of CRE, but other multi-drug resistant infections and bacteria, and what we can do to incentivize pharmaceutical companies to develop and make more antibiotics, diagnostics companies to come up with faster and better ways to identify these organisms and bacteria more quickly, and how we can better provide care for patients with infections caused by MDROs. So here in Chicago, as a result of a legislative mandate that required hospitals to screen their ICU patients for carriage of methicillin-resistant Staph aureus, investigators here at Rush started something called the REALM project. So it stands for Regional Evaluation of a Legislative Mandate. And this is a voluntary network of hospitals in the city of Chicago, acute care hospitals that have more than 10 ICU beds, and all long-term acute care hospitals in Cook County. So investigators go out every 6 months and conduct point prevalence screenings of all of the patients in the facility at one given day, and we screen them for rectal carriage of CRE. We began screening for KPC only in 2010, and then in 2013 we expanded our screening to screen for KPC as well as NDM. What we found as a result of that first initial survey when we were looking for just KPC, when we looked at the 24 acute care hospitals that were participating in that survey, 15 short-stay acute care hospitals out of 24 had patients who were rectally colonized with block KPC-producing bacteria. So out of about 910 patients, only 30 in these acute care ICUs were rectally colonized. In contrast, when we went to the 7 long-term acute care hospitals, or LTACHs, all 7 facilities had patients who were rectally colonized with KPC-producing bacteria. And it was 119 patients out of 391 in total who were surveyed. So roughly about 3% of adult ICU patients versus 30% of LTAC patients. So it's a 9-fold increase in the prevalence of KPC-producing bacteria found in patients with rectal colonization between these 2 different facilities. And that was very telling and striking to us, and that really is why we thought that LTACHs would be a facility where we would need to really intervene if we were try to get a handle on the spread of these organisms and these bacteria on a more regional level. So we're not really so much concerned about the facility level, but we're concerned about how these bacteria are spreading in the region. I don't know if many of you are familiar with long-term acute care hospitals, but these are specialty facilities that are designed to provide care to patients for an extended duration of time. Their average length of stay is 25 days compared to a short stay hospital, which is about 5 to 6 days. And these are patients who are, medically complex, and they often have long-term chronic illnesses. These patients require mechanical ventilation, long-term multiple antibiotic therapy, and they have very complex wound care needs. So these are very, very sick patients. And this is a figure from the Medicare Payment Advisory Commission. They publish this report on a yearly basis, kind of showing you how Medicare money is being spent. So in 2012, there were 420 LTACHs nationwide that received Medicare payments, and Medicare spent about $5.5 billion in 2012 to provide care and reimbursement for these services. And I should apologize, I was not paying attention to my Q&A box that was flashing at me. Dr. Rode is on this webinar, and he's been sending me some messages, so I'm sorry I wasn't addressing those. But Dr. Rode does a lot of work with MRSA, and he's seeing that prevalence of CRE is really paralleled to MRSA, and I agree with that. Here in Chicago, we don't screen our LTAC patients for MRSA as part of the realm surveillance, but I do know from my own experience that these LTAC facilities do have high rates of MDROs in general. And yes, I agree. EVS has long complained that there are a lot of problems with the duodenoscopes and getting them adequately cleaned. And we'll try to pay more attention if you guys do have questions that pop up, so please do send them. Okay, anyway. So when we look at these LTAC patients, and there have been a lot of studies, and I really couldn't just pick one to highlight them, but a lot of authors and researchers liken LTACs to being this perfect storm. You're putting the sickest of your sickest patients in a facility, so these medically complex patients with a lot of comorbid conditions, obesity, diabetes, high blood pressure, wounds, infections, heart failure, respiratory failure, all of these chronic and severe medical conditions. And because these patients are so sick, many of them have central lines, Foley catheters, many of them are on mechanical ventilation. So there's this very high rate of medical devices that are being utilized. And because, again, these are the sickest patients who've had a lot of antibiotic exposure in the LTAC setting, in the acute care facilities, and they have repeated exposures at multiple levels and repeated visits to various healthcare facilities, this really leads to this convergence of these risk factors that makes LTACs really the perfect place to breed, I think, the next superbug. Which is why, again, when we looked at how we could try to best come up with a plan to affect and control the spread of KPC and other CRE in the Chicago region, we really felt LTACs were an area where it was going to be very crucial for us to develop an effective infection control bundle or regimen, because these patients, as they get worse, they tend to cycle back to the acute care facilities because their medical needs change. They require different levels of care. They may return to the LTACHs, or as they get stable and progress, they return maybe to their homes or to a skilled nursing facility for extended durations of nursing care. So these are patients who are cycling in and out of the healthcare system at various different levels. So why is it important now talking about the lab? Why do we have to distinguish the mechanism of carbapenem resistance? And that's really because if you rely solely on your MIC result, it doesn't tell you what the mechanism of resistance might be. It's just going to tell you that, yes, this isolate is resistant or it's susceptible to the drug. Now, some resistance mechanisms, as I said, KPC and NDM are on plasmids. Those are the ones that are an infection control emergency. Those are the ones that we need to identify quickly to isolate these patients, put them on contact precautions to try to minimize the spread of these organisms. Other carbapenemases, like the SME enzyme, SME, is a chromosomally encoded carbapenemase that's found exclusively in strains of Serratia marcescens. Because it's on a chromosome, it's not an infection control risk. We don't need to necessarily isolate these patients because this mechanism, this carbapenemase, isn't going to spread. So really knowing what your resistance mechanism is really going to help guide your next steps. And that's why, the laboratory, the clinical microbiology lab, plays such an important role in the identification of these patients and how your facility can then handle them as a result. So in 2010, the Clinical Laboratory and Clinical and Laboratory Standards Institute, revised their breakpoints for the carbapenem antibiotics to allow facilities to try to detect carbapenemase-producing bacteria a little more easily. So what you see on the left are the previous breakpoints. You can see these breakpoints were much higher. Susceptible to imipenem was less than 4, resistant was greater than or equal to 16. Once they were revised, those breakpoints were dropped by about 2-fold dilution. So susceptible now to imipenem is an MIC of less than or equal to 1 microgram per mil, and it's resistant if it's greater than or equal to 4 micrograms per mil. So that was a way to try to help labs detect these organisms because they were sliding through with these high MICs. Automated systems weren't detecting them. They weren't calling them as carbapenem resistant or carbapenemase producers. So there was a lot of unrecognized or undocumented CRE that was occurring in clinical isolates. So there's a lot of different tests that laboratories can utilize to try to identify. I'm only going to talk about a few. The first is the modified Hodge. I think it's the one that people are most familiar with. In the modified Hodge test, we're actually taking our test isolate, our organism, and we are using it in conjunction with a susceptible strain of E. coli. We're using this ATCC 25922. It's a pan-susceptible E. coli that should be susceptible to all antibiotics when it's tested. So you make a 0.5 McFarland, standard in the microlab. You make that of your susceptible E. coli, and then you make a 1 to 10 dilution in normal saline, and you streak it to a Mueller-Hinton plate to create a lawn of confluent growth. And then you drop your carbapenem antibiotic down on your plate. It's best to utilize meropenem as your antibiotic of choice. And what you do is, after overnight incubation, of course, the antibiotic is going to diffuse out from that disc into the agar and the surrounding area. You take your isolate that you suspect may produce a carbapenemase, and you're going to streak it out away from the carbapenem disc towards the edge of the plate. After overnight incubation, what you're looking for is this zone of growth of your E. coli, which really serves as this indicator. if the test isolate that you're testing produces a carbapenemase, that carbapenemase is going to be present in the surrounding agar, and it's going to hydrolyze any of the carbapenem antibiotic around that slant, that streak of your test isolate. And that is what's going to allow the E. coli to kind of grow into this kind of indented shape, as you kind of see. It almost looks like a Pac-Man to me. So he's kind of where there's growth of your E. coli. In contrast, here on the right, you see the same test isolate's been struck, but you have a nice, even circular zone of inhibition. So this test isolate did not produce a carbapenemase. So this is your modified Hodge negative organism. And on your left is your modified Hodge positive organism. The Hodge test is really easy to perform. Unfortunately, it lacks specificity. It is very poor. It's a poor indicator or poor test to try to screen for Metallo beta-lactamases like MDM. And like with all culture-based methods, it does require an overnight incubation, so it takes a lot of time. Another culture-based method, this one is designed to kind of overcome those barriers of the Hodge test for MBLs, is to utilize an E-test strip that has on one end a carbapenem antibiotic alone, which you can see here on the right, and then on the left is a carbapenem antibiotic meropenem with an inhibitor, which is what the I indicates on your strip. So again, E-tests look at a density of antibiotic gradients, typically increasing in a doubling dilution. So the presence of a Metallo beta-lactamase is indicated when you have a reduction of your Carbapenem-MIC alone by greater than or equal to 3 doubling dilutions of your carbapenem antibiotic in the presence of EDTA, which is your inhibitor. So it's on the side, but I've written, so the MIC of the carbapenem with EDTA is 0.032, and the MIC of the carbapenem alone of this organism is 2. So the ratio is of the carbapenem to the carbapenem and the inhibitor is about a 63-fold increase in the dilution. So this indicates presumptively that this isolate is producing a Metallo beta-lactamase. Now, because it's phenotypic, it does require confirmation, but it's a good screen. Again, it's very sensitive, only works for MBLs. It's very easy to perform, but again, like with culture methods and the modified Hodge, it does suffer an overnight incubation, so it gives it a little longer of a turnaround time. Now, the Carba NP test is another phenotypic method. Now, this method can identify carbapenemases, and it actually can differentiate between 3 carbapenemases, your class A, class B, and class D. This is a phenotypic method that looks at the acidification of the acidification reaction based on the hydrolysis of imipenem alone and in the presence of tazobactam and EDTA as inhibitors. The Carba NP test was actually included in the 2015 updates of the CLSI M100 as a test that labs could utilize to screen for carbapenemase production. I like this test. It's actually very quick. You can get results in as little as 15 minutes, but you do incubate these tubes for a maximum of up to 2 hours. The problem with this test is that you're looking for a change in color, and the changes are reds and oranges, or dark red and light orange. So it's color specific. It relies on a visual interpretation. These colors don't last for very long, so you have to read them at these set points. And if you try to read them beyond 2 hours, the test isn't valid. It's nice so you get you can get the results in the same day within a few hours, but there's some difficulty in trying to interpret it. Now, I've read that there are some commercial manufacturers that are trying to commercialize this make the colors a little more stable, and make it a little easier to perform. But as of right now, it is a very much a manual process, and you have to make up all of these hydrolysis solutions and buffers the day that they're going to be used, because they're not very stable. So more complex to perform, but it can differentiate, and you get the results the same day, but there are some drawbacks. Chromogenic media, this is an area that I would like to see more being done by some of the manufacturers. Most of the chrome agar plates for KPC or other CRE right now are research use only. The problem with these is, unlike our chrome agar plates for MRSA, which are kind of a diagnostic definitive and approved for their use, most of these, or at least the ones that I'm familiar with, again, are research use only. They haven't been approved by the FDA yet here in the US. And they are not, they don't provide a definitive ID. They use a chromogenic substrate and then a carbapenem in the agar base, but Carbapenem-resistant organisms that are resistant to carbapenem antibiotics, either through AMP-Cs or porin changes, can grow on these agars and produce these pigmented colonies. So there's several studies out that have looked at them with varying levels of sensitivity and specificity. So not quite as good as MRSA. If you use them, they really have to be confirmed with some type of confirmatory assay. This is something that's fairly new, the Rosco disks. These are antibiotic impregnated disks. They're much larger than the typical Kirby-Bauer disks, but they are utilized kind of in conjunction with, you have a standalone carbapenem disc, and then you have carbapenem discs that are supplemented with various inhibitors. And this is a way to differentiate between different mechanisms of carbapenemase resistance. I realize the figure might be a little hard to see, but what you're doing is you take your isolate, again, a 0.5 McFarland, streak it overnight onto a Mueller-Hinton agar and drop these various discs. And you're looking at changes in zone size between a carbapenem disc alone and a carbapenem disc with a particular inhibitor. So these Rosco disc kits can be utilized to differentiate between KPC, NDMs, and other Metallo beta-lactamases, OXA-like Carbapenemases, and then AMP-C producers. So in this particular diagram, we're looking at the difference between a carbapenem antibiotic alone, which is this disc kind of in the bottom on the middle, with a zone size of about 26 millimeters, and we're comparing that to the zone of inhibition around a disc with 2 inhibitors. And this phenotypic pattern and these changes in the zone size between the carbapenem disc alone are strongly indicative of, in this case, an AMP-C producer. So this was done by one of our research fellows and published, presented last fall at the ID Week meeting in Philadelphia. Now, the best test that you could do to look for carbapenemases are molecular detection methods, so doing some type of PCR assay. Here we're looking for the detection of the actual carbapenemase gene. So most of these assays, if they can be done real time, would be obviously the fastest turnaround time. So we're looking at primers to bind to a template, a bacterial genome around your gene of interest, and then utilizing sequence-specific DNA probes that have fluorescent tags labeled to detect fluorescence on your real-time analyzer. The pros about PCR is that it's obviously very rapid. You can get results in usually right around an hour or just under an hour, but these are very expensive. Most of these assays have been laboratory developed and validated on their own. The CDC has published and released their multiplex KPC NDM assay protocol that can be run on an ABI 7500 real-time instrument. And I've listed the reference there for you. They describe their procedure. They give you their primer and probe sequences. So it's just up to you if your facility would like to move to PCR and have the capability. But again, it's only been validated, their procedures validated on the ABI 7500. I am familiar that a few diagnostic companies are looking at platforms that have the ability to directly detect some carbapenemases direct from positive blood culture bottles. Both the BioFire has a blood culture ID panel that can detect KPC, maybe NDM. I know KPC target is in there. And then the Nanosphere Verigene also has a similar panel that can detect KPC directly from positive blood culture bottles, which is great, but unfortunately, again, those are only approved for use in positive blood culture bottles and no other specimen types. I'm briefly just going to talk about strategies that the CDC recommends. These can be found and outlined in their toolkit for control of Carbapenem-Resistant Enterobacteriaceae. Hand hygiene is number one, making sure your healthcare workers are performing hand hygiene both before and after patient contact, and constantly educating them on why we have to perform hand hygiene. Contact isolation, so getting these patients isolated and in contact precautions to try to prevent the spread. Getting rid of medical devices when they're not needed, cohorting patients as appropriately, antibiotic stewardship. And again, the lab plays a role, because if the lab doesn't communicate that these organisms are out there, then these are going to spread. And then active surveillance and CHG baiting. So active surveillance, really, these are your gut bacteria. So the rectal isolates have been shown to be the most sensitive in finding and recovering KPC positive, KPC producing Enterobacteriaceae. You can also look at combinations of rectal and inguinal skin sites, because again, if patients are stooling, there can be a lot of stool in the inguinal area, so you can also recover these organisms there as well. So why active surveillance is important? Again, this is going to let us know who has KPC. If we do active surveillance, we can identify these patients, and then we can separate these patients and establish either KPC positive or KPC negative cohorts to prevent the transmission. And in Illinois, our communicable disease code was amended, and we are healthcare facilities and laboratories are required to notify the state of all Carbapenem-resistant Enterobacteriaceae isolates that have been identified in your facility per patient for every admission. So we have to report the first CRE isolate per patient per admission. And this began in November of 2013. Briefly, because I'm running out of time, I just want to go over CHG bathing, Chlorhexidine Gluconate. This is a topical antiseptic that is very powerful, and it reduces skin bioburden and has been utilized for probably about 50 or 60 years in healthcare settings. This is showing how applying CHG onto patients' skin sites and achieving a minimal inhibitory concentration of CHG greater than 128 micrograms per mil on the skin leads to an overall reduction in your relative risk for having KPC positive bacteria that can be recovered from your skin sites. And as my colleague, Dr. Bob Weinstein, always talks about, a clean axilla is a happy axilla. So this is showing you a diagram of a skin culture from an axillary site on a patient in an LTACH, both before and after a CHG bath was given. So before, the plate is just covered with bacteria on the left, and on the right-hand side, you can see immediately after that CHG bath, there was virtually no bacteria detected. So again, CHG has been used for many years in healthcare settings as a way to prevent bloodstream infections and to reduce skin burden. I'm not going to go over this slide. I know the slides are going to be posted as a resource, but this was kind of clinical culture results of our intervention, looking at how our bundled intervention had an effect on reducing the colonization and infection of KPC positive isolates in blood cultures and all clinical cultures and blood culture contamination data. I really want to get to at least one or two of these. This is kind of an interactive session. So I have a quick case here. There was a 51-year-old or 52-year-old male nursing home resident who was bedridden with a G-tube, a Foley, and a tracheostomy, and a multi-drug resistant Klebsiella pneumoniae was isolated from a sacral wound culture. Here is the susceptibility reports in the antibiogram. And this isolate was also tested by the modified Hodge. You can see his isolate is this one up here in the upper corner. So kind of based on what we've learned today, what carbapenemase do you think is most likely present? Again, this is going to be another poll question. Do you think it's the KPC carbapenemase, NDM, OXA-48, or are you unable to determine what the enzyme might be? So just take a few quick, 15 seconds or so to think about that. Okay, let's go ahead and close that poll and see what you guys thought. And then I will explain kind of what the carbapenemase is. All right, so 71% said KPC, and you are correct. So let's take a quick close look, and we'll see why why it's KPC. So if we go back and look at the susceptibility profile, as you look at this, it was tested by PCR and was block KPC positive. If you look at your beta-lactam antibiotics in the panel, you'll see that all of the beta-lactams that are tested are resistant. In addition, the Hodge test, as you can see here, if you can interpret it, there is growth of your E. coli indicator organism around the strain that you're testing. So again, the modified Hodge test is most often and most sensitive for KPC carbapenemases. And then we did a PCR test and found KPC. I will go through, we'll do one more and we'll have to skip the third one, but you'll see it in the handout. This was a 65-year-old female patient who had traveled to Pakistan to visit her family 3 months previously. She's now been hospitalized with respiratory failure secondary to an exacerbation of COPD, and she's been placed on a mechanical ventilator. She had a positive sputum culture, fever, and white blood count. Grew out an E. coli. Here's the microscan panel that you can see here. So we're going to push out just the same question again. So if you want to take just a quick look and think about it. Here's your modified Hodge test. And in this instance, the patient isolate, I'm sorry, is right here on the left. And your poll question might be covering it up, but your Hodge test will say is negative. So what carbapenemase is most likely present? Is it the KPC, NDM, OXA-48, or are you unable to determine? So again, just take about 10 or 15 seconds and we'll go through that question. Okay, So here, very good, awesome. NDM is the correct answer, which most of you did get. So I'll tell you real quickly. This one, it's a little hard to look at probably because it might be a little blurry, but it also unfortunately is using the antibiotic codes instead of their full name. Here we did a multiplex PCR. It was KPC negative and NDM positive. And again, your phenotypic clue at how you can look at this is if you look at your beta-lactam antibiotics, so they're kind of split across these two rows, you'll notice that all of them are resistant. Meropenem is flagging as intermediate, but intermediate is not susceptible, so we would kind of lump that in as an indicator. Your imipenem is being reported as susceptible, even though the MIC is 4, and that's just because the vendors have to utilize, the instrument vendors have to utilize the FDA breakpoints in their reporting, not the CLSI. But your clue really is aztreonam. Aztreonam remains susceptible. It's the only beta-lactam antibiotic that is susceptible of the carbapenemases. When everything else is resistant, that's when you really want to think NDM. I won't do the third case. It was here it is. Again, you'll have the slides with you. I will be heading over to the online learning community after this. I'm sorry I didn't leave too much time for questions, but I will be over there and I'm happy to answer any questions. And it also, my email address is on the title slide. If you have questions about anything, please don't hesitate to email me. I'm happy to answer them if I can. Thank you so much again for the time and opportunity to present, and I am sorry that I went over.
Thank you for that informative presentation. I would like to thank our sponsor, ASCLS, for making today's educational webcast possible. Please make sure to head to the community of learning following this presentation to have the opportunity to engage in live chat with Nick Moore and your peers as well, and get some answers to your burning questions in regard to Carbapenemase-producing GNB, the nightmare bacteria here to stay. Today's webcast will be available for on-demand viewing through October of this year. You'll receive an email from LabRoots alerting you when the webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.